Dear students, you are most welcome in this class. Today is 11th February 2022. Time is about 6 p.m. So today we are going to discuss the topic nitrogen transformation in rice fields. So we have already witnessed or seen some electrochemical changes in rice soils, some chemical changes, some changes in redox potential, and also some physical and biological changes. So mainly the deficiency of oxygen in paddy soil is responsible for most of the changes. Whatever changes we observe in properties of paddy soils, they are mainly because of flooding or submergence resulting due to shortage of oxygen in the soil. That leads to several transformations, several changes in nitrogen also. In nitrogen, you will find variety of forms in nitrogen forms in paddy soils. You will also see several processes. Number of processes are happening in paddy soils. Of course, in aerobic soils also, those processes happen, but some are at very slow pace and some are at very faster pace. And also you see the differences in decomposition product. When you add any organic matter to the soils, whether if it is aerobic soil or if it is anaerobic soil, so you will find different end products. Some products may be common, but some are different. For example, under aerobic conditions, most of the carbon is lost in the form of carbon dioxide. Under anaerobic conditions, some carbon is lost as methane, and remaining carbon remains in soil, but in other forms like organic acids, alcohols. So the organic matter is not fully decomposed. It is partly decomposed. It is partly fermented. So less energy is produced, and the substances which could have produced more energy are still there in the soil, in paddy soil. However, in aerobic soils, such kind of products, organic materials convert directly to CO2 and give or release more energy than in anaerobic conditions. Also, you have fewer species of bacteria under anaerobic conditions. So variety of transformation are happening. Variety of nitrogen products are made during this transformation that we are going to discuss. Before we go into the details of this subject, I would like to ask you, first question is, what is reactive nitrogen and what is non-reactive nitrogen? Can you tell me? Reactive nitrogen and non-reactive nitrogen. Yeah, Dhanavat. You come forward yourself. If you know, you say, yeah, you reply it. Otherwise, you can say, we don't know. It is not necessary that everything is known to everyone.
Don't know, sir.
Don't know. Okay. Reactive nitrogen is a kind of nitrogen that is able or capable to react with other elements, other compounds, and can make new compounds. That is reactive nitrogen. And non-reactive nitrogen is the nitrogen form that cannot react with other elements or other compounds to form a new compound. For example, N2. N2. So this N2 is considered as non-reactive. It does not react directly at its own. As you see, nitrate, your ammonium, nitrite, there are many active forms of nitrogen that can take part in reactions directly. So most of the nitrogen, this element, this gaseous nitrogen, N2 is considered as non-reactive nitrogen. You also know about immobilization, mineralization, volatilization. I assume that these terminology are known to you. Now, in brief, you can see that nitrogen has several roles in plant. It is essential for synthesis of DNA, RNA, chlorophyll, and very important part of proteins. So nitrogen is an essential element and it is necessarily required by all the plants, whether they are higher plants or lower plants. Forms and processes of nitrogen differ between submerged and aerobic soils. So some forms may be common, but some forms may differ in submerged and aerobic soils. Basic information about nitrogen, its forms and processes. Now we will see what are different forms of nitrogen and what are the different processes that change these forms. So in the right side, just you see this table, main forms of nitrogen in soil and their oxidation states. So except N2 in this column, and also RNH3. RNH3 is your organic form. Other forms all are reactive forms of nitrogen, which are in oxidized state or reduced state. And you can see the oxidation state of nitrogen, how much variability it has. As you must see in case of carbon also. Carbon can have variety of oxidation states starting from 0. So here you can see in nitrate, it has +5 charge. Oxidation state means 3 into 2 -6, -6 and 1 charge is there. So -5 and therefore N is +5. Similarly, NO2, it has +4, NO2 minus, NO2 ion, it has +3, NO +2, valency of nitrogen or charge of nitrogen, N2O +1, and N2, it has 0 oxidation state. In NH3, it has -3, NH4 -3, and RNH3 also, it has -3, this nitrogen. So you can see the oxidation state of nitrogen. Now, as you know, it is very, very essential part of proteins, enzymes. It is necessary for life. And it is the nutrient which is the most limiting for rice production. And not just rice production. In India, we have over 50% soils which are deficient in nitrogen. So nitrogen is the most limiting nutrient in crop production. And it is found in all type of animal waste. Nitrogen contents may range from 0.5 to 2% in animal waste. So what is the range or what is the nitrogen content in your soils? Average nitrogen content in soils. Can anyone tell? Yeah, any idea, any guess? What is the total nitrogen percentage in soils, in aerobic soils, cultivated soils? Yeah, Rawat. Any guess?
No idea, sir.
Anyone?
Sir,
Anyone?
1.5.
1.5. So it varies from 0.04 or 0.05 to somewhere around 1% or maybe less than 1%. So it is the amount of nitrogen in cultivated soils. Now, what we see nitrogen around us, most of the nitrogen is in atmosphere, which is made up of 78% nitrogen. And by chance, this is non-reactive. It does not react. That is why it is as such. If it was reactive, then you will not find 98% in atmosphere. It could have come to the soil, it could have gone to the oceans, and there was no N2 in the atmosphere if it was reactive. Therefore, it remains as such in atmosphere. However, much energy is required to break the triple bond between atoms of nitrogen within N2. So if you want to make it reactive, then you must break this bond. This triple bond, triple bond. So how this triple bond can be broken? So it can be broken by energy, or it can be broken by some microbes, or it can be broken by some enzyme. What is that enzyme? Enzyme that can break down this triple bond in N2.
Dinitrogenase.
Nitrogenase.
Dinitrogenase.
Dinitrogenase. I know about nitrogenase, but I don't know about dinitrogenase. Is it new name for nitrogenase or is it different enzyme?
Sir, something different in the molecular form like FPMO and FPFMO complex.
Okay, because till date I know that it was nitrogenase, but you are saying dinitrogenase, then we need to check it. And if you are sure, then we can accept it. Otherwise, we need to recheck it. I heard it.
No, sir, I am not sure. I am listening only.
Okay, no problem. So I think it is nitrogenase, and we can check it. Most living organism cannot use N2 from atmosphere as a source of nitrogen. So it is only few bacteria or some fungi that can absorb or that can directly take up this N2 and convert it into other forms or organic forms. And then those organic forms can be broken down and then they can release reactive forms of nitrogen. So it is just kind of cycling of N2, atmospheric N2 through microbes. So we already know that how much important it was to manufacture ammonia artificially. So in 1900, two German scientists, Haber and Bosch, they developed a process to convert N2 from the atmosphere into ammonia. So they were the first to synthesize this ammonia at large scale for manufacturing of fertilizers. So in this process, N2 is reacted at elevated pressure and temperature. You need high temperature, high pressure, and you also need hydrogen gas. So nitrogen and hydrogen can be combined at high temperature and high pressure to produce ammonia. And the carbon dioxide can be taken from methane, or there can be some other hydrocarbons from which we can take hydrogen. Hydrogen can be taken from methane or it can be taken from other hydrocarbons like naphtha. Naphtha can be used to get hydrogen, and this nitrogen can be reacted with hydrogen in 1:3 ratio, I think, and then you can get ammonia. So this was a wonderful discovery and they received Nobel Prize also. Now, coming to the field, paddy field. In paddy field, what is happening? This is very simple diagram. You can see different layers, the water layer on the top, and water layer, you can see volatilization of ammonia is through this water layer because this ammonium is converted to ammonia. We will see in detail this volatilization. For the time being, you see that volatilization happening from the surface of the soil. From the surface of the soil, ammonia will be released and passing through the water, and then it can come out to the atmosphere. This is, and you can see some oxidized zone is there where you get nitrification. So nitrification happens in the oxidized zone, and then you are seeing the reduced zone below the soil surface, below the this oxidized layer, and you see mineralization, immobilization, denitrification, all are happening below the or in the reduced zone. You can see below the oxidized zone. And for details, you can see here, this picture is much better explaining the same. So you can see it is, you can start from anywhere in this picture. We can start from water. You are seeing the water, and below the water, you can see the aerobic soil layer in the picture. And aerobic soil layer, you can see ammonium. So this ammonium can come from urea, or this can come from decomposition of organic matter, or it can come from any source. It can come from ammonium that is fixed in the clay lattice. Sometimes ammonium fixation happen in the 2:1 type of clays, and that ammonia can also be released. And so there can be many sources of this ammonium. And this ammonium can go to several routes. It will have many fates. So this ammonium actually has 4 or 6 fates. It can go into 6 different routes. I will discuss later what it can be done from ammonium. But here you are seeing that this ammonium is converted by nitrification into nitrate. first to nitrite, then to nitrate. And you can see this ammonium is can also come from the reduced zone. Reduced zone, it can come upward by diffusion. So you can see this ammonium is coming, and where from this ammonium is coming in reduced soil, reduced zone, you can say it can come from mineralization of organic matter, or it can come from deep placed urea fertilizer. Sometimes urea fertilizer are applied at deep placement in the reduced zone. Then this ammonium can come from there also. And from there, it can diffuse and reach the plant roots for uptake. Uptake of ammonium can be there by the rice plant. And this ammonium can also be immobilized, and it can convert to the organic nitrogen. And this organic nitrogen can come from the biological nitrogen fixation. There can be two different sources here. It can, this organic nitrogen can be made by ammonium, or it can come by biological nitrogen fixation, organic nitrogen. And there can be some other sources of this organic nitrogen also. We will see in detail. But they are in equilibrium, this organic nitrogen and this ammonium. They are in equilibrium, and mineralization, and it can go to mineralization, or it can go for immobilization. Several factors will affect this mineralization. Now you see, go to the aerobic layer, and you can see here also surface applied urea fertilizer. Surface applied urea fertilizer can very easily volatilized in the form of ammonia. Under certain condition, high temperature, high pH, it can be easily converted into ammonia. That is about 50% losses of urea can occur due to ammonia volatilization. Particularly when you applied ammonia or ammonia forming fertilizer on the surface of the soil. So such ammonia volatilization losses also occur. You can see now, come back to again to ammonium, and nitrification is happening, converted to nitrite and nitrate. And nitrate can go for downward diffusion. Downward diffusion, it can go to leaching, or it can be denitrified. It can be denitrified because it is in reduced zone now. So here its oxygen will be used by microbes, and it can be denitrified by microbes and converted to N2, N2O, and they escape to the atmosphere. So nitrate leaching, nitrate are lost by leaching and also by denitrification. So these are very simple processes happening in paddy soils, and I think you are aware of all these. It is just, I'm refreshing your memories so that you get more confidence on this. I've tried to summarize the whole picture, whole thing, whatever is happening in the paddy soils into these three rectangles. So you can see nitrogen in soil. You see the central rectangle. Nitrogen in soil, if we see, it comes through two sources. One is external application, one is internal nitrogen, inherent nitrogen. Some nitrogen is already there in the soil, and some nitrogen you apply from outside. So this is total nitrogen in the soil. You apply from outside and the native nitrogen. This, whether you apply from outside or it is there in the soil, then you will have several processes like mineralization and immobilization, volatilization, etc. So several processes will happen in the soil. Now on the left side, you can see what are the inputs of nitrogen, how nitrogen enters the soil. So nitrogen can come through organic sources, whatever organic sources you have, manures or cakes or whatever you want, you can add. Fertilizers, nitrogenous fertilizer. It can come from irrigation water, some nitrogen in some cases, lightning from atmosphere, and biological nitrogen fixation, etc. So these are the major inputs reaching the soil. Some are organic in nature and some are inorganic in nature. Now see the output, mineral nitrogen. So whatever organic sources you have, they will undergo mineralization. If they are going mineralization, they can produce ammonium and nitrate. Ammonium and nitrates are produced, are the final product of mineralization. And what is their route? They can go to crop harvest or crop uptake, absorption by the crop. Runoff can happen, even erosion can happen. Erosion can take away your ammonium and nitrate from the soil. Leaching of nitrate can happen. In some cases, in soils where cation exchange capacity is very low, leaching of ammonium nitrogen is also possible. You would surprise to note it, but sometimes ammonium leaching also happens besides nitrate leaching. Volatilization in ammonia form and denitrification also happens. And the reactive forms are ammonium may go for volatilization, nitrate for leaching, ammonia lost, N2 in denitrification, and N2O. So these are different forms in paddy soils you can see. Now we will see these things in detail. So nitrogen entering the rice system, as I told you, nitrogen fertilizer like urea is very common fertilizer, ammonium sulfate, NPK fertilizer, many fertilizer. Biological nitrogen fixation is also there in paddy soil by Azolla or Azolla anabina symbiosis or by free living bacteria. Lightning also contributes some nitrogen to the soil. So these are different sources of nitrogen and how it leaves, how it is lost from the soil. So crop harvest in grain and it is removed. I must have told you before that if you harvest 1 ton of paddy grain, you take out approximately 20 to 20 kilogram nitrogen, 20 to 22 kilogram nitrogen per ton rice harvest, rice grain harvest. It includes nitrogen in straw as well as in grain. So suppose you have 5 tons of yield, then about 100 kilogram nitrogen or 100 to 110 kilogram nitrogen will be removed in grain plus straw if you harvest 5 tons of grain. So that means huge quantity of nitrogen is harvested along with the crop harvest. Ammonia volatilization primarily happens for nitrogenous fertilizer, which are applied on the surface. If you applied on the surface, most of the nitrogen or up to 50% nitrogen can be lost through ammonia volatilization in alkaline conditions. Denitrification, and in the reduced zone, you see nitrate reach there, and they are converted to N2 and N2O or other oxides of nitrogen. Runoff means if you get flooding and some flooding can take away nitrogen from paddy field to other areas. Leaching, you know that nitrate can move along with the water and reaches beyond the root zone. Then this leaching can also result in losses. So these are the important losses. So there are two kinds of major forms of nitrogen in the soil: organic nitrogen and inorganic nitrogen. This is true for other soils also. So as you know that most of the nitrogen is in organic form. More than 95% of nitrogen is in organic forms in the soil. Suppose you have 100 units of nitrogen in the soil. So now more than 95% units are in organic form and less than 5% are in inorganic form. So what are the inorganic forms? They are ammonium, they are nitrate, nitrite, etc. So plants take up nitrogen in an inorganic form. Nitrate and ammonium are the main inorganic forms. Nitrate and ammonium represent a very small fraction of total nitrogen in soil. So in total nitrogen of soil, you also have available nitrogen, which includes your nitrate nitrogen, ammonium nitrogen, plus readily oxidizable nitrogen. So there are 3 parts of available nitrogen. So these are the forms. Dinitrogen, we already discussed ammonia, nitrate, nitrite, and ammonium. And ammonium and ammonia are in equilibrium. Organic nitrogen, CnH2 in ammonium amino acid forms, nitrous oxide. So there are different forms. You are quite aware of these. And reactive nitrogen, I have already told you, most forms of nitrogen in the environment are reactive. They react chemically with other compounds and or biologically with other organisms. They are reactive nitrogen. Non-reactive nitrogen is your N2. Let us see different processes which are responsible for conversion of these forms. So processes are immobilization, mineralization, ammonia volatilization. You are very well clear about these. Immobilization, plant available form of nitrogen, are used by microbes and becomes temporarily unavailable. Mineralization, organic forms of nitrogen are transformed into a plant available form. And ammonia volatilization is urea fertilizer, converts to ammonia gas and is lost to the atmosphere. This ammonia volatilization can also happen for the native nitrogen. If it is there in the, if it is near the soil surface and it is in the ammonical form. So it has come from diffusion from the reduced zone to the aerobic zone. Nitrification and denitrification, you are aware of these terms. So these are 5 important processes that are responsible for conversion of nitrogen into different forms: immobilization, mineralization, volatilization, nitrification, and denitrification. So you are seeing all these in pictures. Another important process is biological nitrogen fixation. So where microbes convert N2 into a form plants can use. So this N2 can be converted by microbes, maybe rhizobia, which are known as diazotropes. Diazotropes, or it can be converted by azospilum, clostridium. Variety of organisms are there that can break the triple bond and convert this N2 into amino acid and then proteins. And when such materials are added back to the soil, they can be decomposed and produce nitrogen in nitrate or ammonical forms. So this is how it takes time to get nitrogen in mineral form from N2 to NO3. Ammonium fixation is also there. I think you are aware. I repeat that you can go to Brady's book, Nature and Properties of Soil, where you will find the detailed information of nitrogen fixation. Similarly, potassium fixation is there, K fixation. So K fixation and ammonium fixation, they happen together in chelates. You have a number of chelate structure in soil: malt, morillonite, kaolinite, elite. Variety of clay minerals are there, and some of them fixes nitrogen in ammonium form in their interspaces or interlayers, where potassium is also fixed. For details, you must read this subject from Nature and Properties of Soil. Potassium fixation is also same, means entry of potassium ion into the interlayers of clay minerals, that is K fixation. Similarly, ammonium fixation. So if you want to release ammonium from the fixed clay minerals, then you need to apply potassic fertilizer. If you apply potassic fertilizer, then ammonium will come out from the clay interlayers. So this is very important. You can read. Now, organic and inorganic nitrogen. Nitrogen transition from an organic form to a plant available form through the process of mineralization. And what are the processes that affect the availability of nitrogen to rise? So microbes are very, very important. You need adequate growth and population of microbes if you want to mineralize the organic matter or organic nitrogen in soil. So of course, under these paddy soils, you get anaerobic and aerobic microbes, and they do mostly anaerobic respiration or fermentation kind of thing. So microorganism population grows to make use of this food supply. If you are adding some fresh organic matter into the soil, irrespective of the soil, the microbes population will increase because they are getting some food. So how these microbes are doing their job? So what they do, these microbes release certain enzymes into the soil. they are not directly attacking the organic matter. They attack organic matter by releasing certain enzymes, and those enzymes are involved in the reactions. Otherwise, these enzymes can also come from the plant roots. Certain plants can release certain enzymes into the soil. They can be involved in mineralization processes or other kind of processes in the soil. So these microbes are very, very important. So via enzymes, they are involved in the decomposition process or any reactions. Whatever is mediated by microbes, it is through enzymes. But microbes are not the only source of enzymes in the soil. Other sources could be plants, could be weeds. Now, immobilization, like rice plants, microbes need nitrogen for their life cycle. Because any microbe, whether it is bacteria, fungi, actinomycetes, they also have DNA, they also have RNA, they also need nitrogen, they also need protein, they also require carbon, they also need phosphorus. So most of the nutrients which are required by higher plants are also required by lower plants. And nitrogen is the most important. So therefore, they consume the nitrogen which was there in the soil solution. Soluble nitrogen can be removed by microbes, and that is why this nitrogen depression periods comes and plants may suffer, particularly when your CN ratio is wide. If CN ratio is narrow, then you will not have this immobilization issue. So this is your immobilization. Similarly, mineralization. Microbes break down complex nitrogen compounds in organic matter. The final nitrogen product is ammonium. I already explained you how this mineralization happens in soil. Now, CN ratio is very, very important for mineralization or immobilization. I recommend Nature and Properties of Soil. You must read organic matter chapter. So CN ratio in soil, you can see it is very, very important. So this is the ratio of carbon and nitrogen in the soil. So normally in the cultivated soils or arable soils, it is 8:1 to 15:1. This is the range, and the median being 12:1. In India, we say 10. So you can say 10 to 12:1. Ratio is generally lower for subsoils. In subsoil, you have proportionately more nitrogen than carbon, or rather compared to the surface soils. So in surface soil, CN ratio is wider. In subsoil, CN ratio is bit narrow because organic matter is less or organic carbon is less, but nitrogen may reach through leaching of nitrate or sometimes ammonium. So in a given climatic region, little variation occurs in CN ratio for similarly managed soils. So CN ratio does not change very fast. It changes very slowly. If you have similar kind of climatic region and little variation in CN ratio, there will not be any variation in the CN ratio if you have similar management practices. In more severely leached and acidic A horizons, in humid regions, CN ratio is as wide as 20:1. In forest O horizons, you must understand O horizon, it is 30:40 ratio, CN ratio. However, CN ratio of soil microbes is less variable than in plant tissues, but also much lower. CN ratio ordinarily falling between 5:1 and 10:1. The range is 5:1 to 10:1. Among microbes, bacteria are generally richer in protein than fungi and consequently have a lower CN ratio. Bacteria have lower CN ratio, fungi have wider CN ratio of 10. 10:1 for fungi, 6:1 for actinomycetes, and 5:1 for bacteria. So we continue the carbon to nitrogen ratio. Now you understand that fungi have 10:1 CN ratio and your bacteria have 5 CN ratio. So very narrow CN ratio. They are full of nitrogen. these bacteria. Means you can see 10 means the 90 part is carbon and 10 part is nitrogen. And 5 means, how to say is carbon to nitrogen ratio. 5 part carbon and 1 part nitrogen. 10 part carbon, 1 part nitrogen. So you can see these bacteria are very rich in nitrogen. So they need nitrogen for their food. So now carbon to nitrogen ratio, it influences the decomposition rate. The decaying, the mineralization is affected by this CN ratio of the organic material. So if it is wide, if it is more than 25, 25:1, as may be case of many cereal straws, the nitrogen in organic material is not enough to support the decomposing microbes. So when they start decomposing, some of the nitrogen will be released from the organic matter, but that nitrogen may not be enough for the microbe. So they need some external source. So therefore, they take up nitrogen from the soil solution or available nitrogen. So they use surrounding nitrogen, and therefore some depression, some shortage of nitrogen may be there. But what happens if your CN ratio of the organic material that going to be decomposed in the soil is narrow? Narrow here means less than 20. 20:1, which is represented by certain legumes. So enough nitrogen is in the organic matter to support the decomposing organism. Means they will produce nitrogen of their requirements themselves or own selves. Nitrogen from organic material can be released into the soil and be available for growing rice plants. So under both cases, in one case, there may be shortage of nitrogen. In other case, there may not be any shortage of nitrogen for the crop plants. But both the process, whether it is decomposition of wider CN ratio residue or narrower, ultimately nitrogen mineralization or net gain in nitrogen will be there. Means in the end, you will get more nitrogen what was there in the soil, more mineralized nitrogen what was there in the soil, mineral nitrogen. Now you see very interesting picture from Brady book. So you can see here this picture on x-axis, you have time after placing residue on soil. Means they are decomposing the soil, these crop residues. Decomposition starts from 0 day up to 128 days they have tested, and mass remaining means what percentage of organic material has been decomposed and what is remaining. So the section or part of the organic material that is remaining is shown on y-axis. So you can see there are 3 different lines. The orange line, the topmost line, is showing you a material which is a cereal material, cereal rye. It is a crop, grass in America. So CN ratio is 87, very wide. And see the green line, it is having some legume, which is vetch. VECH, vetch is a legume. CN ratio is 10. And what they did, 50% organic material from the vetch and 50% from the cereal. So this dotted line, red dotted line, is for that. So you can see with the passage of time, the mass left for the green, for the legume, is very less, or you can indirectly say the decomposition rate is very fast. And for C is carbon is to nitrogen ratio 87, the decomposition rate is very, very slow. So you can see the difference. And when you get mixture of cereal and legume, then the decomposition rate is intermediate. Now see, this picture is also very important for you. Influence of CN ratio on residue decomposition we are dealing with. So in the first picture, you can see here the CN ratio is high, more than 25 has been taken in this case. And you can see on x-axis, it is time. And on y-axis, there are relative values. On y2 axis, it is CN ratio on the right side, 0, 20, 40, 60. This is your CN ratio. So you can see on the left side, organic material is added at the arrow. You are seeing the arrow. And here you see some soluble nitrogen level in the soil. So you can see the soluble nitrogen level is high, is sufficient, looking to be sufficient. But when you have added residues, organic residues here of wider CN ratio, arrow in the place of arrow, then you can see suddenly there is deficiency of nitrogen. This is known as nitrate depression period. And here, if you see the other two lines, the red lines, one line is denoting to the CN ratio. The CN ratio, you can see it is written in the on the line CN ratio. So you can see in the beginning, the CN ratio was wide when you added organic matter above the arrow, and then it is slowly decreasing, and it has come below down to 20. Initially, it was more than 20, but it has come down to 20 or less. The other line, the third line, is showing you microbial activity. So microbial activity is indirectly measured by CO2 evolution. More CO2 evolution means more decomposition. More decomposition means more microbial activity. So you can see when you have added organic material, the microbial activity is very fast. And then after a certain period of time, it starts declining. So in the end, it declines. Now you see the soluble nitrogen level in the soil. In the beginning, you can say that the levels are low. But on the right side, when you see this shaded area, the mineral nitrogen has increased. Now you go to the other figure on the lower side. Now same thing, you can see here. First you see there is no nitrate depression period, because here the residues near the arrow were having narrow CN ratio. Narrow CN ratio, hardly any depression was there in nitrogen. Very small depression is there. And practically you can say there is no nitrate depression period here. And you see continuously the levels of nitrogen are increasing. This shaded area is showing you this nitrogen levels. And see about carbon dioxide or microbial activity. Microbial activity in the beginning, it is very high, and then it starts declining after a certain period of time. But you can see the peak, means here the microbial activity is very high, but for a very brief period. And then suddenly it drops. In another case, it drops slowly. It drops slowly. So you can have variety of interpretation from these pictures. The details of these pictures are explained in Nature and Properties of Soils. Now, net mineralization of nitrogen, the differences between amount of nitrogen mineralized and immobilized is called as net mineralization. Means how much is the gain of mineral nitrogen. In submerged soils, the total nitrogen immobilized and total nitrogen mineralized are typically less compared to the aerobic soil. Fewer microbes, because of fewer microbes are present in the submerged soil, those present operate at lower energy levels. So net mineralization is usually higher for submerged than aerobic soils. Following decomposition, now you can see the decomposition. Main key points in this: microbes responsible for decomposition require nitrogen for their growth. So nitrogen is must, and these microbes can consume the nitrogen and immobilization will happen. But later on, when deficiency of nitrogen happens in the soil solution, then it can get decomposed or it can get mineralized. Now, suppose you are adding urea as a source of fertilizer, then on the surface, not as incorporated, then you will have ammonia volatilization losses. So what happens, this is about urea. Suppose you are adding urea as a source of nitrogen or any ammonia forming fertilizer. So after urea is applied, it reacts with water and the enzyme urease in a process called hydrolysis. Ammonia is the product of reaction. I assume that everyone knows the hydrolysis of urea reactions and the nitrification of urea. So when urea is applied in submerged soil, the process is complete in a few days, maybe 10 days, 12 days, the urea will be hydrolyzed. Now, fate of ammonium, as I promised before, now you can see what will happen to this ammonium. So it can go by, it can be lost by soil erosion, it can be lost by runoff, and besides that, there are 6 more fates of this ammonium. It can go for immobilization by microbes, removal by plant uptake. Anamox means the anaerobic oxidation of ammonium in conjunction with nitrate to produce N2O gas. So this is new term, anamox, means the anaerobic oxidation of ammonium in conjunction with nitrate to produce N2O gas. So volatilization after being transformed into ammonia gas. Volatilization is clear to you, nitrification is clear to you, and fixation or strong sorption in interlayers of certain 2:1 clay minerals. So this could be the fate of ammonium. Now, anamox, if you want to know more details, so information is given very nicely in book, page number 615. You can see it. Now, ammonium and pH, because urea is hydrolyzed, ammonia is produced, or ammonia can come from reduced zone by diffusion to the aerobic layer or to the top of the soil. So there is some relationship with this ammonia and pH of the soil and pH of the water also. So ammonia, NH4 and NH3 are in equilibrium. The ratio of ammonium to ammonia gas is affected by water pH. At neutral pH, ammonium is strongly favored. At neutral pH, ammonium is strongly favored. As pH increases, the amount of ammonia gas relative to ammonium increases. Means if you increase the pH, production of ammonia will happen and it will be lost. For detailed reaction, go to Brady book. Water pH and volatilization. You can see the relationship of soil water pH and relation. See this picture, effect of water pH on conversion of ammonium to ammonia. So two lines are there. One is for ammonium, other is for ammonia. On x-axis, you have pH. On y-axis, you have percentage distribution. So you can see at pH 5, there is very little volatilization losses. At 6, 7, very little or insignificant. But when you increase the pH after 7, there is increase up to 8. And after 8, there is too much increase in ammonia volatilization losses. And same time, it has inverse relationship, opposite relationship. So in the beginning, ammonia and ammonium were in happy position. Both were happy, they were in equilibrium, but suddenly this ammonia losses increased. Then the quantity of ammonium reduced. So you can see from this table. Now, water pH and CO2. Now see the relation of CO2. We have seen for ammonia, water pH changes with amount of CO2 in water. The CO2 affects the pH of the water. As CO2 goes up, pH goes down. You know this carbonic acid is produced and hydrogen ion reduce the pH. Algae growing in the rice paddy influences CO2 in water. Photosynthesis by algae uses CO2 and water pH goes up during the day. Respiration releases CO2 and water pH decrease at night. So this is simple explanation for diurnal changes in pH of the water. This is most pronounced in the week after applying fertilizer. So you can see daily rise and fall of water pH in a rice paddy caused by changes in CO2. Now, we continue with ammonia volatilization. You can see this reaction, how it is happening. And once ammonia is converted to ammonia gas, it can be lost into the atmosphere through volatilization. This is major cause of nitrogen loss, can be as high as 50%, and wind accelerates the transport of ammonia from water surface and increases the loss of nitrogen because it will reduce the concentration around the soil. Now, how to reduce the ammonia volatilization? The best and simplest strategy is to mix the urea into the soil or mix the fertilizer into the soil. Other options are that you can apply urease inhibitors, or some other strategies can be adopted. Placing urea in soil rather than incorporation of urea is the best way to reduce ammonia volatilization losses. So key points are that ammonia volatilization is most significant in the week after applying nitrogen fertilizer. A week, because it takes time to get nitrogen from urea in ammonium form. So almost all the ammonia all the urea, or say more than 90% of urea, will produce ammonium after 1 week or 10 days, depending upon several things, but most of the urea nitrogen will be converted to ammonium. Then you will have great accumulation of ammonium ions. And under those conditions, you will have very high volatilization losses. So ammonia volatilization is greater than when rice plants are small. Less shading, because in the beginning, in the initial stages, less shading from rice plants favor algae growth. Photosynthesis by algae can lead to increased water pH. Rice plants require less nitrogen during first weeks after establishment. Requirement is also low, delaying nitrogen application until crop demand for nitrogen increases helps minimize ammonia buildup and loss through volatilization. Nitrogen forms now, up to now, we have seen the fate of ammonium. Now let us concentrate on nitrate, because these are two important mineral forms in soil. They also exist in paddy soils. So nitrogen forms and oxygen zones. So what can happen when nitrogen is exposed to aerobic environment and anaerobic environment? Be able to describe the two nitrogen process, we'll see. So now see ammonium and aerobic soil. So you can see here that water layer is there, aerobic layer is also there, and anaerobic layer is there. So three layers are visible, and this ammonium form is present in anaerobic soil layer. This ammonium, I have shown you where from this ammonium has come. But this ammonium from reduced layer can go up, and it can be oxidized in the aerobic layer, or it can be oxidized in the rhizosphere. Region around the roots, rhizosphere, where oxygen is there. So ammonium is the primary form of nitrogen in anaerobic soil. Zones of submerged soil where O2 can be present are two zones only, rhizosphere and the aerobic or thin aerobic layer. Ammonium can move by diffusion into soil zones with oxygen. It can go up. Now the nitrification will happen around in the aerobic layer as well as in the rhizosphere. So ammonia is in the presence of oxygen may be changed. I think you know how this ammonium is converted to nitrite by nitrosomonas bacteria, and then nitrobacter to then nitrite to nitrate by nitrobacter. And in soil, you can remember this nitrite form is nitrite NO2 minus 1 is very, very unstable. It does not remain in for a long time. If it remains for a long time, it is very, very poisonous, very, very toxic to the plants as well as to the microbes. So nature has made a provision that I will not allow you to stay long. So suppose within seconds, this NO2 is converted to nitrate. It does not take much time. So when you go for analysis of soil, you will find very traces of NO2. You can find nitrate, you can find ammonium, but you cannot detect NO2 in soils. So negligible quantities of NO2 are there in the soil. So these processes are both part of nitrification, this first reaction and second reaction. And you know, enzyme is monooxygenase, I think, monooxygenase released by nitrosomonas. Now you will see one more term, code nitrification. Code nitrification. It is a related microbial process by which nitrite in the soil solution is reduced to N2O and N2 gases. N2O is nitrous oxide by such nitrogen compounds as ammonium. So ammonium can reduce this. Ammonium or hydroxylamine carried out by bacteria, streptomyces and fungi, Fusarium oxysporum. For details, I have given you the reference. So this may be a new subject for you. Code nitrification, you know, and then what is code nitrification? You can learn in detail, very half-page information on this in this book. Now, fate of nitrate. So it can go for leaching, it can go, erosion and runoff. Besides this, there are 6 more mechanisms by which it can be lost, or by which it can move from one form to another form, or one place to another place. So first fate is immobilization by microbes, removal by plant uptake, microbial reduction to NO2, followed by conversion along with ammonium by NMOx to N2O gas. Denitrification can happen. It is converted to N2 or N2O, and dissimilatory reduction by microbes to ammonium. Microbes can directly reduce it to ammonium, and leaching, loss of groundwater. So there can be 6 routes for this nitrate in paddy soils. Denitrification, you can see in this picture how this denitrification is happening. So in denitrification, you can see this nitrate from the right extreme. Nitrate is leached down from the aerobic layer to the reduced zone, and in reduced zone, it is oxygen is used by microbes. You can see the detailed procedure in the book, and NO3 to NO2, and then NO2 to N2O or N2. So you can have losses in the form of N2 or N2O from the paddy soils. This is denitrification. This is mostly mediated by microbes, but it is also mediated. It is also a chemical process, but which is not which does not have very significant value. So denitrification is mostly a microbial process. Release of nitrogen gas. N2 is the primary product of denitrification. N2O can also be produced depending on conditions. It is potent greenhouse gas and it destroys ozone. Nitrification and denitrification can result in loss of nitrogen fertilizer applied to the farmer. So ammonium to nitrate and nitrate can be lost. So it is clear to you. So key points here are: avoid buildup of excess nitrogen. Apply nitrogen to meet crop needs, means you can go for a split application of nitrogen. And as far as possible, in rice case, it should be placed in the reduced zone, if possible, by mud ball technique, or there are some urea supergranules. Of course, success is less, but in countries like Bangladesh, they have adopted this technology in rice, and they have machine, they have techniques, and they are using it. This urea super granule, they have applicator, they have machine that can place the urea super granule in the reduced zone of the soil. So it has found some practice in Bangladesh. Minimize the amount of nitrogen remaining after the crop season. Alternate wetting and drying of rice paddy can result in more nitrogen loss through nitrification and denitrification. Of course, the mineralization can be faster, but all nitrogen produced by mineralization may not be used immediately by the crop. Therefore, leaching can occur in alternate wetting and drying. Means alternating wetting and drying can result in increased availability of mineralization of the nitrogen, but at the same time, it can also increase the losses by denitrification and leaching. Normally, leaching losses are very minimum. Leaching losses are very minimum in rice. Main losses are by denitrification and volatilization. And if it is applied, say basally, means what I mean to say, if you continuously keep your soil flooded, then the leaching losses are very minimum, very insignificant, because whatever nitrate is produced, that is produced only in aerobic layer, and that can be used by a plant that can leach down. If it is leaching down, then it can be denitrified. So whatever nitrate is reaching to the reduced zone, it can be denitrified, and very little nitrate is left for leaching. So in submerged paddy soils, losses by leaching are minimum compared to denitrification and volatilization. Many times students consider that leaching losses should be more, but that is not the case. Leaching losses are the least. The most important loss is your by volatilization, second is denitrification, and then your leaching. Biological nitrogen fixation, most of you are aware of this. Symbiotic and asymbiotic kind of relationship exists here. So diazotropes are important. While nitrogen in the atmosphere is mostly unavailable to plants, a group of bacteria called diazotropes convert N2 gas, means N-fixing bacteria or organism are your diazotropes. So there are certain facts that on a global scale, the amount of nitrogen gas fixed by diazotope is almost equal to the synthetic fertilizer. So large quantity are coming from nature, free of charge. That is your biological nitrogen fixation. In tropics, lowland rice yielding 2 to 3.5 tons per hectare have been maintained for centuries with bacterial nitrogen fixation, because in those days there were no fertilizer. So what people used to do, they used to take only one crop, and then for 6 months they used to keep land fallow, and at the same time you can have some biological nitrogen fixation in the soil, and also you can get some nitrogen from the lightning also. And sometimes people used to graze their animal also in those fallows. So overall, the soil fertility sustaining system was very good that time. So what I mean to say here is that biological nitrogen fixation is very, very important, particularly for rice. So there are a number of options for biological nitrogen fixation in rice. So you know about cyanobacteria or blue-green algae. These are single-cell organisms living on the surface of water or plants, and they can also live in association with this fern. What is the name of that fern? Single-cell organism living within submerged soil and the root zone of rice obtained. There are some free-living bacteria like your Azospilum, Azotobacter. They can be in the aerobic layer, aerobic regions, and Clostridium can fix nitrogen in anaerobic region. And you have Azolla, that fern is Azolla, Anabina association. Some species of Azolla fern grow in association with Anabina azoli. So you are aware, and they can produce about 20 to 40 kg nitrogen per hectare. So much of nitrogen can be fixed. It is common, or it is used in low-lying areas of Bangladesh, in Vietnam, Philippines, China. Many countries are taking advantage of this association. However, in India, it is not so common. Only some wild algae grow in our fields. Can require additional phosphatic fertilizer. This susceptible to insect pest disease. So it is subjective to several conditions. Growth can be affected by, and of course, these legumes are important. This rhizobia is a kind of diazotope fixing nitrogen in symbiosis. And examples, you know, what are the pulses and all legumes, they can fix nitrogen, but they can be taken in the rotation with the rice crop, or their residues can be added into the soil. Some green manures can be practiced before rice. In summer season, this green manuring is practiced, and that can save 60 kg nitrogen. 60 kg nitrogen can be saved. You need to apply 60 kg less nitrogen if you are taking rice after the green manuring of sesbania or any other legume crop. And not just green manuring, but you can take green legumes in the rotation. They are also helpful. So rotation with rice, legumes are useful. And overall, we need to reduce the losses, leaching losses, runoff losses, ammonia fixation, etc. So for ammonium fixation, you can go through the book. I have already told you in detail. You can get the knowledge on ammonium fixation. It is common to aerobic as well as anaerobic soil, but it is more so in anaerobic soil, because here production of this ammonical form of nitrogen is more compared to aerobic condition. In aerobic, it is quickly converted to nitrate. So accumulation of ammonium happens in paddy soils. So these are the summaries. I will share this presentation so that you can read the summary of this lecture. It was a bit lengthy. I tried to cover most of the things, and now your questions are welcome.